All right, let's talk about classifying viruses, which goes back to taxonomy. So we've talked about this first week of class. We talked about taxonomic classifications, which also the naming system is nomenclature. Taxonomy is categorizing the different living things. So when we talked about this before, we were talking about living things, and we talked about how the way organisms are named reflects their ancestry. We figured that out from DNA sequencing, right? It used to be looking at physical features, but it's much more accurate to look at the DNA. So just like you can do, a paternity test or ancestry.com to figure out who you're most related to through your DNA, you can extend that out to other species. So the more similar the DNA, the more closely related. And so then we name organisms to reflect in a way that reflects their ancestry. So even when you just have the name of something, you can quickly figure out, because knowing something's ancestry gives you a lot of information about it. So this same system we apply to viruses as well. And the reason is that they evolve. So how, you might think, well, how can something that's not alive evolve? Well, all evolution means, so there's the evolution technically means change over time. And then there's biological evolution, which is a change in trait frequency over time, right? So if you're just thinking of it as change over time, well, everything evolves then, right? Living and non-living things, everything changes. It's not like, life is more dynamic than inanimate objects. And viruses are in that gray area between life and non-life. But even when you're talking about evolution from a biological perspective, viruses are not alive because they cannot metabolize or replicate on their own, and they're acellular, but they still have a genetic message that gets copied and changed, copied, changed, copied, changed. So you still see this evolution or this change in genetic message over time. And not only that, but that genetic message is expressed as traits. Now, viral traits are very simple. They have to do with the proteins only. There's pretty much just, the virus is just genetic material and protein, as you know. So the changes can be, the traits can be what spike proteins for attachment to host cells does it have. So when we're talking about evolution of coronavirus, you all know that coronavirus was once a virus that attacked another animal, maybe a bat, and now evolved to attack humans. So how did that happen? What traits changed? Well, the trait that changed was the attachment proteins, right? So it has these spikes that it uses to attach to a host cell. Those spikes only used to bind to ACE2 proteins in bats or some whatever animal it ends up being. And then it mutated. It changed shape slightly because there was a change in the DNA, which resulted in a change in the protein. And the change in the protein ended up being such that it could attach to ACE2 proteins in humans and not just bats anymore. So that's, and I say bats because we're just guessing at this point where it came from, but the other coronaviruses that have infected the human population often come from bats, not always, but often. So that's just a guess, but you get the idea. So, that's how viruses end up jumping species, as their attachment proteins. Capsid proteins can change. Enzymes for DNA replication could change. So whatever protein, whether it's enzyme, attachment protein, structural protein, whatever protein it is, those are the traits that a virus has, and they can change over time because of mutations to their genetic message, their DNA or their RNA. So viruses were once upon a time classified by symptoms because they were too small to see, and we didn't have DNA or RNA sequencing technologies. So you just had, the only way to classify them was what symptoms do they cause in the patient. But it turns out there's a lot of closely related viruses that can cause very different symptoms, and there are a lot of viruses that are completely unrelated that can cause the same symptoms. So symptoms and do not necessarily match up with relationships, but with genetic relationships. So now relatedness is determined by genetic sequencing. That's the most accurate way to do it. We can just take a virus, sequence its DNA or RNA, figure out what it's related to. And there's a lot of usefulness in this. So first of all, you can figure out something about the virus if it's genetically related to other viruses we've known, that can help us treat it. So if you know, okay, this new virus that came in, it's a type of flu, it's a kind of influenza. So what methods, what medicines do we have that we use for influenza? What treatment practices do we have for other kinds of influenza, and can they be applied here? Can the vaccines that we've used in the past be applied here, right? So that's one way that taxonomy is useful to us. This classification is useful to us of viruses. But the other reason it's useful is when a new virus emerges, in terms of emerging infectious diseases. Excuse me, I have to sneeze. Excuse me. When a new virus arises and we're finding it in these pockets all over the globe, and we're trying to figure out where is it, where isn't it, how is it traveling, how is it being transported, the best thing to do is take a bunch of samples from people and figure out whose virus is most related to whose. So for example, let's just say this virus we found across the globe, we want to know where it came from and its path of travel, how it's traveling. Is it traveling through animals? Is it traveling through migrating birds? Is it traveling through mosquitoes? Is it traveling through infected travelers? What is the method by which it's moving around? So what you do is you take all people, there's people who you know are sick with this virus, you take viral samples, you sequence their genetics, and which virus is most related to which will tell you the path of travel. So for example, if you take, in the terms of coronavirus, if you, I don't know the path of coronavirus travel, I'm sorry, I haven't kept up with the exact path, but I'm just saying as an example, you take this virus, let's say it started in Wuhan, China, and then you have it in Sweden, and you have it in Durango, and you have it in Texas. So so then you take all of samples of the virus from all of these sick people, and you sequence their DNA, and you find that the virus in Colorado and Wuhan is closely related, and you find that the virus in Sweden and Wuhan is equally related, but they're not related to each other. So you can say the two closest relationships are Sweden and Wuhan, Denver and Wuhan, and then Texas is most similar to Denver. Well, that would tell you that maybe there was a branching. So maybe some infected travelers went to Sweden, and at the same time other infected travelers went to Denver. So you had this split, simultaneous split, and then the infected travelers from Denver brought it to Texas, but not the ones from Sweden. Okay, so so what I'm saying is you can track pathways because the viruses that are most closely related to each other are the ones that were in the same place most recently. All right, so those are reasons. So it's the same taxonomy that we use for living things, but they're in a different domain. So we talked about the 3 domains of life. There's a 4th domain that's for viruses, and it's not a domain of life. It's a domain of this unliving, this non-living microbe. Mutation and adaptation. So you did an activity before the exam on HIV, and so I want to talk about that a little bit because I'm sure there are questions. So HIV has a rapid mutation rate, and eventually it evades most effective therapies. So a person with HIV, and I want to distinguish HIV from AIDS. HIV refers to the virus. AIDS is the disease the virus causes. So you can have HIV without AIDS if the virus is lying latent. Once it starts being expressed, you will come down with AIDS. Okay, so AIDS is the set of symptoms. HIV is the virus itself. So HIV is replicating in the bloodstream. A patient takes an antiviral for it. It works. It knocks back the virus, but then the virus evolves. There's these mutants that are drug resistant to this drug, so then it starts coming back with the drug-resistant mutants. The drug no longer works. You have to use another drug until the virus becomes immune to that drug, and so on and so forth, until a patient might go through every drug available on the market until none of them work anymore because the viral load is now resistant to all of the therapies. Some mutations will always arise that provide therapy resistance. Why? Because, so in the case of HIV in this video, and I'll link to the video again, HIV and most viruses replicate incredibly rapidly. You might have thought that bacteria replicated rapidly. They double in number every hour. That's pretty typical for bacteria. It's not even doubling. Viruses they don't use doubling time. They generate massive, they don't go from 1 to 2, 2 to 4. One gets inside a cell, makes hundreds, thousands of viruses, and they all burst out at the same time. So they have this rapid replication rate, and it's a matter of minutes, seconds. It's just so fast. It's orders of magnitude faster than bacteria. So you get mutations really quickly with viruses, in part in all viruses, because they're replicating so quickly. Some viruses mutate more rapidly than others because some of them have really inaccurate replication equipment. Okay, so for example, there's some viruses that use our equipment to make more DNA. They're DNA viruses that make more DNA, and they use our cells' DNA synthase, which has proofreading capabilities and doesn't make mutations very often. But then you have RNA viruses that bring their own enzymes that read RNA to make more RNA. Instead of reading DNA to make more RNA, they read RNA to make more RNA. Those virally provided enzymes are really inaccurate most of the time because they don't have proofreading capabilities. Retroviruses, they're RNA viruses that reverse transcribe back to DNA. So they come as RNA, they reverse transcribe back to DNA and insert that DNA into your chromosome. Well, the virus has to bring an enzyme that can read RNA and make DNA from it because the host cell doesn't have that. Again, doesn't have any proofreading capabilities, so it makes mutations, makes errors all the time. So HIV is a retrovirus. It doesn't copy itself too well, and it replicates really rapidly, so you're going to have massive numbers of mutants all the time, and some of those mutations are going to just happen to provide drug therapy resistance to the virus. So that's the whole point of structured treatment interruption. So the idea with structured treatment interruption is that in structured treatment interruption, you have a patient whose virus is resistant to all the therapies available on the market. And so to try to control evolution, so the idea is that you want to control the evolution of the virus instead of fighting evolution, which always seems to be working against us with microbes, where we use scheduled treatment interruption to control the evolution of the virus in the direction we want it to go, which is to control the environment, the natural selective pressures to cause the virus to evolve back to wild type. And the way you do that is by removing the drugs. So the thing is, as we discussed with penicillin resistance in your activity, there's a trade-off to these mutations. Yes, they provide drug resistance, but they also have some cost. So there's a protein that you target with the drug. The protein has a job for the microbe. The protein when it mutates, it is drug resistant, but it's also not doing its original job as well. So there's a cost. So when there's no drug there, the mutants don't do as well because they've got a mutant protein that's not doing its original job as well as the original. But that mutant protein, it's not doing its job as well, but it's also resistant to drugs. So you add a drug, now the resistant mutant takes over. If you take the drug away, the resistant mutant isn't going to be favored anymore. The wild type, the drug susceptible one, is going to be the one that does better without the drug. So that's the idea behind structured treatment interruptions. Now they're dangerous. Structured treatment interruptions can be dangerous for the patient because when you go back to wild type, the drug susceptible variety, those ones are much more efficient killers of cells because they all have the original proteins that did their job the most efficiently. Their proteins are the optimum at replicating, at attaching to host cells, at invading host cells, whatever it is that those proteins do that the drug was targeting. The original versions that are drug susceptible are also much better at their original job. So if you drive the evolution back to wild type, it's dangerous for the patient because their viral load will become much more deadly. But the benefit is it's more drug susceptible again. So once you get the wild type back, you can hit it hard with drugs, knock the viral load back again. Okay. Now, you might wonder, how come the viruses never completely disappear? How can you get the wild type back if the patient had the normal wild type, drug susceptible type at first? Supposedly, when they took the antiviral, they killed off all the viruses. All the wild type viruses that were drug susceptible and left only the resistant mutants behind. So how's the wild type coming back if you eliminated it with drug therapy? Well, the answer is that HIV is a retrovirus, and retroviruses are never gone from your body. They insert their DNA into the host chromosome, and you will learn about that in the upcoming lectures. But what they do is they have their viral DNA, they insert it into the human chromosome where it stays forever. Forever. We do not have a way to cut open the human chromosome, take out the viral DNA again, and sew it back together again. Fuse the ends back together again to heal around it. We can't do that. So retroviruses genetically modify the human cell by putting their own genes into the human genes. So they are never gone. So that's another piece of that video that you watched before the exam. Is HIV is special because it's a retrovirus. Once it's in your DNA, it's always there. So I'll link to this. So how can we use what we know about evolution to effectively treat patients infected with HIV? I'm going to post this video again if you didn't watch it for the extra credit before the exam, because here it becomes necessary for you to know it.